Have you ever wondered where horses originated from? You might think we can figure the answer out purely from archaeology, but we would like to discuss another way, through DNA.
We will first focus on bow tie settlements in Kazakhstan. It is said that some of the earliest domestication of horses occurred here.
Start out with this graph. So we have time right here, and we have 4 separate lineages. As in the article, we learn that the bow tie settlements we were looking at over here, starting with this horse in the beginning, over time, their remains were found in Kazakhstan, and those actually went extinct. So this lineage stopped right here during time. Then also we have another lineage that was found in Russia, which also went extinct. We have our third lineage in Portugal and Spain, also went extinct. And when we finally arrive at the Arabian horse over here, which we found to be the true ancestor of our modern horse, it keeps going over time and far exceeds and is the last remaining lineage in the horse's ancestry. So let's put on our lab coats and be scientists and think about, how does this all work? How do we use DNA to track lineages? So one method is using Y inheritance, which tracks the passing of the Y chromosome from the father to the son. Let's talk about how that works. So let's start out with the basics here. For example, we have a father and a mother who have a son and a daughter. The father will have his X chromosome and his Y chromosome right here. The mother will have 2 X's, as you can see. In the father's case, we know for sure that he will pass down his Y chromosomes to his son. That can happen in any case. And we also know that he will for sure pass down this X chromosome to his daughters. However, on the mother's side of the family, we don't know whether this X chromosome was passed down to the son or whether this one was passed down to the son. Same thing with the daughter. It could be the green or the red. We just don't know. So that's why you can't actually use the X chromosome in lineage tracking, because we're not sure if the mother will pass down this one or the other. So therefore, the Y is much more frequently used. Things get even more complicated when we talk about crossing over. So during cell replication, the DNA duplicates itself, forming sister chromatids on each analogous chromosome. So you have here an analogous pair, which during crossing over, they will lie on top of each other and exchange DNA. So as you can see, once crossing over has occurred, you have a little bit of the red chromosome in the blue and vice versa. When we have a male, however, things are a little different. So in a male, you have an X and a Y chromosome, which here you can't just start crossing over because they're different sizes, they don't match. They don't have equal segments of DNA that you can just switch. So the difference is that this Y chromosome over here will remain the same. The only difference is that it will accumulate mutations throughout generations, demarcating the passage of time, and therefore it becomes a very useful tool in lineage marking.
So right now we're looking at the sequence on the Y chromosome of our original Arabian horse, and we have 4 offspring of his. And over the years, we see mutations form on that Y chromosome. And so there's positive and negative, and one of our negatives that we found is called hyperkalemic periodic paralysis, and that's actually from a C to a G substitution. And then through the years as well, there's the positive, and that is where we get faster and stronger horses, so we continue to breed them. So our modern horse is actually a lot more stockier and taller than our original Arabian horse.
In conclusion, by tracing the mutations in the Y chromosome, we know that none of the lineages found throughout Eurasia contributed to the modern horse. In fact, it was the Arabian lineage.
Ha 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 ha!